You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Erik Gunther. And today I have Erik Gunther. He's an associate research scientist in neurology at Yale, and he's working on Alzheimer's and trying to assist in the drug discovery, drugs that will actually work for it. So, Erik, thanks for coming.
It's a pleasure. Thanks for inviting me.
Yeah, so tell me, how did you end up in Alzheimer's research in the first place? What spurred you to move in that direction?
Well, I don't know. I'm a neuroscientist, and I've always been interested in seeing if that interest could actually be brought to some sort of benefit. The lab that I'm in actually studies neurodegeneration and neuroregeneration. We started off on spinal cord injury and repair of spinal cord injury. Then we moved into neurodegeneration. And when the lab discovered the receptor for the A-beta protein, the amyloid beta protein that causes plaques to form in the brains of Alzheimer's patients, and most people think it's the reason people get Alzheimer's. When we discovered the mechanism of action for that protein, it suggested a way that we could address it with some sort of pharmacology strategy.
Yeah, I've heard that there's been, 150 different drug trials for Alzheimer's, right, and they've all failed?
Yeah, that is true. In large part, that's thought to be because the behavioral symptoms that are diagnostic for Alzheimer's come late in the disease. It's a progressive disease, it's a chronic neurodegeneration, and it's only when that's fairly progressed, when the neurodegeneration has happened for a decade or more, that you start having these behavioral problems. Your memory starts suffering, and you start becoming demented, disoriented. And so attacking the disease at a late stage like that is kind of closing the barn door after the cows have left. It's much more important, most likely, to address it early on.
Well, what about the model itself? Have there been any rumblings or alternate models instead of the beta amyloid plaque accumulation?
Well, the amyloid hypothesis is the most commonly accepted hypothesis, but we don't know for sure. I mean, we haven't cured it in people yet, and that'll be the bottom line. Most of those drug trials you were referring to were geared against amyloid, the amyloid protein. Again, the reason that they probably didn't work is because that's the trigger. The amyloid protein buildup in the brain is what kicks off all these pathological changes in the brain. And they have kind of a life of their own as they progress. Taking away the initial insult does like getting kicked and having a bruise, right? I mean, if you try treating the bruise after someone's kicked you, it's not going to really help the healing. The idea is to prevent the kicking in the first place, and that's probably going to have the highest prospect for success anyway.
So can you run through for people that don't know, what's the process by which we think people get Alzheimer's? What happens first, and then next, and next, and next?
Yeah. So, like you're saying, with the amyloid hypothesis, the thought is that there is an accumulation of A-beta or beta amyloid in the brain. It starts off as a small protein, and some of the interesting work that's coming out now as to why the brain produces that is that this small protein is an antimicrobial peptide. It's generated by the brain to fight infection, or viral or bacterial infection. And after a while, and it works, but after a while, 20 years later, you get this accumulation of that peptide in the brain, and it causes the pathological changes.
Once the peptide is accumulated, how does it affect changes, and what are the changes?
Right. Well, so the changes in the brain, you get neurodegeneration. Ultimately, you get synapse degeneration to start off with. You have impaired communication between neurons. It's mediated by the synapses. We measure that. We can take neurons and culture them in vitro and treat them with this peptide, and we see that their ability to communicate is impaired. And when we discovered the mechanism by which that takes place, we found that we could stop that interaction, and the ability of the neurons to communicate was restored. But after that, in the disease, in human disease, after that that takes place, a synaptic impairment takes place. There's a subsequent synaptic degeneration. So on either side of the synapse, you have retraction of the two components of the neurons that are communicating. And then finally, subsequent to that, you have more downstream pathological changes. You have wholesale brain cell death, neuron death, and also phosphorylation of a protein called tau. That's one of the two hallmarks that you can see in the brain of Alzheimer's patients. That we think that's downstream of A beta, of the A beta action. You were referring to there being different models for what causes Alzheimer's. Some people think that tau is the main bad actor, and that's the initial insult, and the A beta isn't the first one. But I think it's fair to say it's a possibility, but that opinion's in the minority.
Are these changes in the brain happening adjacent to the beta amyloid tissue, or is it all over the brain?
It starts progressively, and it is relatively in the area where you see the plaques. And I don't know how much detail you want to get into, but there's this small peptide, as I was saying, that gets expressed by the brain in order to fight infections, possibly. Then those peptides, they aggregate. They form these small clumps that diffuse around, and then those eventually, we believe, accumulate into the large plaques, which are these really obvious gummy deposits in the brain. And interestingly enough, it's not the initial peptides, not the short length that has the toxicity, and it's not the plaques that have the toxicity. It's this intermediate form that's called an oligomer, this small diffusible aggregate that can move around the brain and bind its receptor to kill the neurons.
Have people sequenced the beta amyloid plaques to see what they look like, their structure? Are they heterogeneous?
The sequence of the peptide is known. That's been known for a long time. But the actual structure of the aggregate, that's an area of study. And our lab is actually investigating that, trying to figure out the structure, the three-dimensional structure of the oligomer, of that intermediate diffusible form that has toxic action on neurons.
Does it appear to have a structure, or do you know if it's homogeneous or heterogeneous?
It is. There are a lot of different structures. Each of those, so it's a heterogeneous assemblage, but each of the structures are likely to be defined. So the single peptide, the short length of protein, the A beta monomer, has a particular sequence, and it's unstructured. So there's no real question about what three-dimensional structure it has. It doesn't really have any. It just sort of moves around in space. But when that aggregates, it's likely that it follows different pathways. And then ultimately, when it aggregates into the plaque, that's kind of a mess. That has certain structures called a beta sheet structure predominantly, but the details of that are also unknown.
At any of the stages, does it tend to create a microenvironment that's different from other parts of the brain, or what used to be those areas of the brain?
Yeah, that's a good question. There have been studies that have looked at the plaques and seen if they are toxic themselves, if there's a greater concentration of the oligomer, the intermediate form, that's given off by the plaque. And by and large, it looks like the plaques are pretty stable, that they don't actually release that toxic component. There are some studies that look at the local effect of the plaques on neurons, on the axon on neurons, and it appears that the plaques interrupt the flow of different parts of the neuron, or that engage in cellular respiration and cleaning up of waste products in the cell. So there is something that the plaque. Yeah.
Do you think they're physically blocking, or are they? Are plaques essentially alive or dead or metabolically active, or what are they like?
The plaques are just gummy goo. They are not alive at all. They are just clogging up the brain. And the actual they do have a physical presence, but it's probably not a mechanical effect. They are probably having some sort of interaction with the axon. Although, that's mostly speculation. That really hasn't been demonstrated one way or another.
Is there any, does the body seem to clear out plaques and then maybe perhaps is overwhelmed by their accumulation? What do we see in normal people?
Yeah, well, in normal people, the plaques don't accumulate. And the plaques in the brain are really not cleared very efficiently. There are cells in the brain called microglia that their job is to clear out those like plaques, and they actively do engulf the A-beta peptide. And so it probably is a balance between clearance and accumulation. But the plaques themselves are fairly resistant to clearance. The peptide and the oligomer are more susceptible to clearance.
Is there an immune system in the brain, or is there?
Sure. Yeah, that's what the microglia.
I don't know much about it, which is funny now that.
Yeah. Yeah, there's definitely an immune system. It's distinctly different from the rest of your body, the rest of your physiology. The immune system in the brain is a little more primitive. The brain is populated by neurons in part, and they are what's called postmitotic. They don't replenish as easily as cells in the rest of your body. So you can't really have a super aggressive immune system clearing out all sorts of cells and causing toxicity. So the brain is immune privileged in that regard. It has a more primitive immune system with microglia that do the clearance.
Have we observed any other coincident strangeness, changes in the blood-brain barrier or effects on parts of the brain that are very distant to where plaques accumulate?
Well, the blood-brain barrier does get compromised in Alzheimer's. Is that what you're asking?
Right. What other things come along with it besides, or in addition to the plaque formation and the neurons dying and neurodegeneration?
And neurons dying. Yeah, I would say that's probably the other main component is breakdown of the blood-brain barrier. The vasculature becomes unstable. And in fact, that turns out to be, people have Alzheimer's and they die of it. And one of the main mechanisms by which they do die is they will have strokes. The vasculature in the brain will become so impaired by just the structural breakdown of the brain that it becomes leaky and there's bleeding in the brain.
Oh, wow. Any other unusual or incidental findings that may or may not be correlated with the buildup of plaques?
Yeah, I'm not sure I know exactly what you mean.
Well, like if I didn't mention it, I didn't know that the blood-brain barrier and the vasculature gets compromised. Are there things like that are either explainable or not explainable that occur?
Yeah, not really. There's hypometabolism in the brain that's kind of an early sign of Alzheimer's, and it's not known at all how that happens. Basically, you have insulin insensitivity in the brain. So some people call Alzheimer's diabetes of the brain, which doesn't really mean much. It just points to the idea and points to the phenomena that there's decreased responsiveness to insulin. So there are metabolic changes as well.
So even the brain becomes insulin resistant, or can become insulin resistant.
Right. And diabetes is a risk factor for Alzheimer's, but there's really not a clear mechanistic relationship.
Well, what happens when you see a brain that is insulin resistant? What kind of changes do you see in it?
Well, you see decreased glucose uptake, essentially, in a PET scan. So, it's if somebody comes into the clinic and and a diagnosis of Alzheimer's is sought, there are a number of things that can be looked at. One is atrophy in the brain, and that you look at the size of different brain regions by MRI and look for degeneration of, for example, the area of the brain that's involved in learning and memory called the hippocampus. Often that will be deteriorated, and the area of the brain around the hippocampus will be shrunken or can be shrunken. And then there's metabolism. You look for with a PET scan, you look for decreased activity, glucose uptake in the brain. There's also a diagnostic technique that's recently becoming more used, where a compound that fluoresces when it comes in contact with plaques is injected into people's bloodstreams, that gets absorbed in the brain, and then that can then be visualized, and you can see if there are actually these glowing spots that correspond to the presence of plaques in the brain.
Have we observed the start of Alzheimer's in patients, and does it seem to correlate with the timeline we established, and have we seen any reversals?
Well, the only real diagnosis that has 100% certainty is Alzheimer's, and then, when you dissect their brain and do an analysis, you can see the plaques and the tangle of Alzheimer's. Some people are diagnosed with Alzheimer's incorrectly, and it turns out to be something like vascular dementia.
Okay.
So it's difficult to answer your question. It's difficult to say, have there been people who recovered from it? Because if someone were to recover from it, you might say, oh, well, they didn't have Alzheimer's. So really the short answer is no. Nobody has recovered from Alzheimer's ever.
But have patients that have Alzheimer's, have they had repeated PET scans to see what's changing and how and the progression? And does that align with what's theorized? And has there been any slowing of progression or reversal of progression, according to a PET scan on some people?
There has never been a reversal of progression, and there's never really been a treatment that has been shown consistently to affect the progress of the disease. There are a couple drugs that are out there that mostly treat the symptoms. They increase the concentration of a neurotransmitter in the brain or something that will kind of like a cup of coffee. It'll boost your brain so that you perform better in the short term, but the actual pathological mechanism of the disease is unaffected. So, even people who have Alzheimer's disease, they often will fluctuate by the time of day or the day. Some days are good, some days they're bad. So there is sort of an inconsistent presentation of the symptoms, but in general, on average, they get worse and worse and worse until they die.
Yeah, the reason I'm asking all these questions is I'm trying to solve the mystery with you right here on the phone. Figure this out.
No, it's great. These are the mysteries that we grapple with all day, every day.
How about sleep? I've heard, I don't know if this is true or not, but supposedly the brain tissues shrink or pull back or condense in a way to allow clearing out of,
Yeah, right. That was a very exciting paper that came out a couple of years ago, I guess, where it was shown that the concentration of soluble A-beta fluctuates quite a bit between waking and sleeping hours. That upon sleep you get basically taking the trash out.
Well, have PET scans been done on, for instance, Alzheimer's patients while they were sleeping versus while them being awake? If that's even possible to observe any differences?
Yeah. PET scans, I'm not sure. I don't know. Could be.
Okay.
I don't know.
Well, maybe if people could be sedated in such a way that the brain clears out the trash, just like when sleeping, to emulate that, and then
right. Just put it in a medically induced coma. Yeah.
I don't know. When people have Alzheimer's, do they tend to have problems sleeping? Does it affect their sleep?
Oh yeah, definitely. It often impairs sleep, and that's one of the recommendations is you engage in sleep hygiene, is you make sure you get 8 hours of sleep, 8 hours plus a night, in order to try and give yourself some defense against contracting Alzheimer's.
Well, what does Alzheimer's appear to do to someone's sleep pattern? They wake up early, or they can't sleep? Yeah,
usually don't. They do wake up early. They get less sleep at night. Their sleep is less. You don't sleep as soundly.
Okay, interesting. Yeah, I just wonder what the correlation is, and if any therapies, again, if the brain changes what it does while you sleep, I wonder if that would be a time for certain therapies that would work better than other times. Because there's more clearance perhaps for,
right.
I don't know,
well,
small molecule drugs to run around in there.
Well, stimulating clearance mechanisms is the idea behind most of those drug trials. And the way they've done it is to generate antibodies against A-beta primarily, so that the body's immune system will clear from the brain. And as I say, these have not been effective clinically, and we don't know why, but we speculate that it's because the disease is just too far progressed. By the time you start clearing out that initial toxic insult of the amyloid protein, the brain is already dying, and there's nothing to be done.
So when people say the studies have failed, does that mean they've like completely failed, or there's been a little bit of an effect, or has it just been a little bit of an effect would be a success. If we could even slow down the rate of progression, that would be a success. All of them have failed pretty conclusively. No change to the rate of progression of the disease. Okay. If diabetes possibly is a risk factor, and if the brain has insulin resistance, have any of these trials controlled for diet? You know, had some folks on a regular diet versus some on a low carb or ketogenic diet, for instance, to see the effects, or controlled for it at all?
I'm actually not that familiar with the literature regarding trials and diet. I know there's epidemiological data that shows that a Mediterranean diet is associated with a decreased incidence of Alzheimer's, but I'm not familiar with actual trials of diet. They may have been done. I'm just not sure.
Yeah, it's kind of mysterious because a separate trial on diet is one thing, but I would think when they're doing these trials, if you controlled for diet, it would make for a better, clinical trial, because that's just one less factor that varies.
Yeah, well,
and it could be a big one.
What. Yes, but I'll tell you, it's a lot easier to design a really good trial than it is to actually conduct one. Getting people to comply with their just taking the drug in the first place is a big challenge. But getting them to change their lifestyle, their eating habits while you're doing it, and especially for something like Alzheimer's, where you need a trial that takes place over many years, and you need thousands of people to participate, actually enforcing something like that really becomes implausible.
Okay. And then I've been asking 8 million questions, but so what is your research focused on specifically?
So, like I say, we found this receptor that's on neurons that the A-beta protein binds to have its toxic effect. And that suggested a site, a focus of action where we might be able to design a drug that would bind to the receptor and compete with A-beta peptide, A-beta protein, so that it couldn't bind the receptor. And that's in fact what we did. We developed a biochemical assay and screened tens of thousands of potential candidates, chemicals, for the ability to block the interaction between the receptor, which is called PRP, and the A-beta toxin. We actually found that the molecule that we focused on, it binds PRP in this active site, in the region of the PRP that interacts with A-beta, in a fairly broad way. It's actually very unusual for a drug. Most drugs are small molecules, and they have a low molecular weight. The one we discovered is a polymer, and it's much higher molecular weight than is typically thought of as being developable as a therapeutic. But we found that when we administered it to mice, we could either inject it directly into their brain, or with a subsequent generation of molecules of that type, we can even administer it peripherally. The initial one we found was a degradant of an antibiotic, a cephalosporin antibiotic called ceftazidime. And we tried giving ceftazidime, just regular fresh ceftazidime, unaged, to mice, and it had no effect. But when it's aged, it degrades, it spontaneously forms this new compound, which is this polymer, this active polymer. And when it's injected into the brain, it rescues the mice. Their synapses regrow, their memories get back to normal as though they never had Alzheimer's. And then this next generation that we invented, we can administer that peripherally. It doesn't get into the brain very efficiently because it's so big and it can't cross the blood-brain barrier very well, but it does enough. It's a very potent compound, and it gets into the brain enough to have an effect, and again cause the synapses to regenerate and the behavior of the mice to recover to normal.
How do you think what is its method of action, you think?
Oh, it blocks the interaction between A beta and PRP receptor. That's it. So normally you have these clumps of protein, right? The soluble aggregates of A beta floating around the brain, and they bind the receptor, and that's how the neurons, when they feel that, they die, or the synapses retract, and over a period of years they die. So the idea was, if you, instead of trying to remove the A beta from the brain, which is what all these trials have done and have all failed doing. Well, they succeeded in removing A beta from the brain, but they failed in actually having a rescue of humans. So we thought, instead of trying to remove the plaque, we can render it inert by preventing its interaction with this receptor. So we leave the structure of the brain intact. We don't worry about the A beta that's there. We apply this drug, and it prevents the toxic action of A beta. It's kind of a novel way to approach the problem.
That's weird, though. I mean, a couple weird things. Why would there be a receptor on neurons for something that's toxic to them? It kind of doesn't make sense why that would be there.
Well, that's not its normal function. I mean, the normal function of the PRP protein is a matter of active study. There are a lot of ideas about what it does. It also happens to be the protein that causes mad cow disease. So when it misfolds. So its purpose is not there to be destructive. Its purpose is there to conduct other things. But when things go haywire in the brain, it starts having a pathological effect.
I've also heard that some. Go ahead.
No, I was just going to say, that we all die for a reason. We all age and break down because evolution wouldn't happen otherwise. You need to get old people out of the way so that they don't compete for resources with the young people to, conduct their lives. So there's really no selective pressure to prevent toxicity of your various aspects of dysfunctional physiology when you're old. It's actually, in some cases, better to get you old and dead to get you out of the way so that the offspring can survive. That's just sort of a philosophical thing about how evolution works and why, why would you have something work in your brain? Why hasn't evolution weeded out these toxic effects? Well, there's no selective pressure to get old people to survive. In fact, it's better to get them out of the way from an evolutionary standpoint.
So no one understands what PRP does or how it functions yet?
Well, there are ideas that it's involved in copper ion binding and toxicity mediation in the brain, that it's an antitoxic function. But like I say, it's still a matter of open investigation.
And this is kind of a crazy idea, but I seem to remember that certain people that are drug addicts, I don't know what drug it was, either cocaine or MDMA or whatever it was, it would cause synapses in their brain to build too many connections and build them as strange spots. Instead of things pulling back and dying, it would actually encourage them to just grow, but kind of grow crazily.
Randomly.
So they had an overload of connections in the brain. Maybe it's crazy, but I wonder if that would maybe help in Alzheimer's. Maybe it would just send things in the wrong direction, but maybe it would help.
Maybe. I don't know about that drug. I'm not familiar with that.
Just because of the method of what it does to the brain. That's what
right.
into my head.
Sure, you want to counter the synapse destruction by giving it a little goose, right?
Probably the wrong goose. I'm just thinking, it's
yeah.
I get into it and try to figure it out.
Yeah, for sure. I mean, that's that of our profession is come up with ideas like that. Well, if this is happening, what about that? And just basically do a thought experiments and then see how they pan out at the bench.
So you said when certain drugs have been able to remove the plaques in the brain, but it had no effect?
Antibody-based drugs, typically. And they work in mice, but they don't work in humans. And again, probably because we're just doing it too late. If they were applied early, and this is the general thinking in the industry now, and there are efforts to try and identify families, and there's a study going on in Colombia where there's a group of families that all have a mutation that give them Alzheimer's very early. So this enables a test of antibodies that will clear the plaque administered even before they get overt signs of Alzheimer's. You know that because you have this mutation, you're going to get Alzheimer's in your 50s or early 60s. So as a result, if they start treating people in their late 40s and then monitor their progression, it's possible to see that if treating the disease early with an anti-amyloid therapy will have an effect. And this is specifically to address the question of whether or not the amyloid hypothesis is correct. Is it really the A-beta peptide that's causing the disease? Again, most of us believe that, but it hasn't yet been definitively proven. And because all of these clinical trials that have addressed the disease late in its progression have failed, they're trying to address it in an amyloid clearing strategy early in the disease to demonstrate that it actually is this timing issue, that if we can address the disease early with an A-beta directed treatment, it will have an effect.
And what was the incidence of Alzheimer's, I don't know, historically, and what is it now? Is it increasing, and what is it?
Well, it's tough to say because people are living with Alzheimer's, but also the resolution of Alzheimer's is getting better. Our diagnostic capabilities are getting better. I mean, now we have these chemical tools, the PET scans and so on that are getting higher resolution. Before it was just, they're senile or they're getting old. They got old-timers disease. It wasn't really the records that have existed previously are not really comparable to the present records. So it's probably relatively the same. It's just a matter of diagnostics, differently effective diagnostics.
Well, what appears to be the rate? What percentage of the population gets it?
Well, so it's the sixth leading cause of death in the U.S. It's if you're over 85, I believe the statistics are that there's a 50% chance that you're going to get Alzheimer's.
Wow.
Yeah, it's tremendous problem. I mean, many other diseases have yielded to modern medicinal chemistry and basic biological understanding of disease mechanisms. People die of most other high-risk diseases, or treating stroke and so forth. But Alzheimer's increasing as a cause of death because we keep people alive long enough to die of Alzheimer's. I mean, it's like 200. Yeah.
What about early onset? Is that when does that happen? What does that look like? And has that been changing, increasing, decreasing?
Yeah. So early onset is a result. So most Alzheimer's disease is what we call idiopathic, which means we don't know what causes it. Really, we don't really know. That's like 95 at least % of Alzheimer's is idiopathic, and there are some genes that will increase your chances of getting idiopathic Alzheimer's. But there is a small percentage of Alzheimer's disease, less than 5%, that can be caused with 100% certainty by mutation in a couple genes. Those genes produce more of the A-beta peptide in the brain and cause you to get plaques a lot sooner. And they're actually in one of the mutations that produces the A-beta peptide. So this is one of the strongest pieces of evidence that is amyloid hypothesis that is the most likely explanation for Alzheimer's disease initiation. That if you get a mutation in the gene that encodes the A-beta peptide, and you get one of these particular mutations, you will get a lot more A-beta in your brain than you would normally by a certain age, and you will get Alzheimer's with virtually 100% certainty. And that's the early onset Alzheimer's. Almost all early onset Alzheimer's disease cases are a result, or most of them anyway, are a result of a mutation in one of the genes that causes an increase in the buildup of the amyloid protein.
All right. So what appears to be the promising future for your work, over the next few years? What do you think, where you're headed with it?
Well, we have a number of experiments that we've published that look at different components of this pathway, right? Downstream of A-beta binding to the PRP receptor, there are biochemical changes that take place in the neuron. And there are ways to manipulate those changes, and we've looked at those either pharmacologically or genetically. And in most cases, have rescued the mice. So basically marching through the biochemistry that's downstream of A beta in the brain that exerts this toxic effect. So it's our hope that one of these focal points of interaction, of intercession, will result in some actual clinical efficacy. We've got most of these targets. I mean, the one that I developed against the PRP protein, that's a candidate. There are more conventional drugs, more small molecule drugs that target some of the enzymes that are downstream of the receptor and activation of the receptor by A beta. Those have worked in mice. And so it's our hope that we can marshal the resources to get some clinical trials accomplished with these various strategies.
It seems to be a lot easier to, improve the lives of mice than people, unfortunately.
Well, it is. You can control the mice a lot more easily. Clinical trials are tough to do, very expensive. And, pharmaceutical companies are justifiably cautious about putting a lot of resources. They only have so much money, even though they've got a lot. And the FDA hurdles are steep. And so it takes a lot of money to conduct a clinical trial, especially something, an indication like Alzheimer's, that just takes so long to evaluate the efficacy of treatment for.
How long does it take to evaluate?
5 years at least, 5 to 10 years to do a clinical trial for Alzheimer's.
And when someone has Alzheimer's, how long do they have it before they pass from it?
It's an average of 10 years since the time of diagnosis to the time of death.
Gotcha. Okay.
Yeah. So, to answer your question a little more definitively about, how long will a clinical trial take, it depends on how well the drug works. If the drug cures you right away, then it's not going to take very long to see a difference between people that are gradually progressing in Alzheimer's and people who have, been cured of the disease. Their memories are going to improve, and the curve is going to go in an opposite direction, and that would be readily discernible. Whereas if you have something that would decrease the rate at which you are getting worse, then being able to separate those curves is going to take longer to resolve. That resolution will take longer. So it's really difficult to give you. I gave you a pat answer of 5 to 10 years, but it really depends on how effective the drug is.
Okay, well, the ballpark works. Well, very good. So what's the best way for folks to learn more about your research and maybe to talk to you about collaboration or ideas?
Well, there's most of our work's published in the scientific literature, so I certainly would encourage people to read scientific journals. To learn more about ours, I suppose they could look at our lab website. It's the Strittmatter Lab, Yale. STRITTMATTER.
Okay. Well, very good, Erik. Well, I appreciate you coming on the podcast, and thanks for sharing your knowledge.
Yeah, thanks. It's a pleasure. Nice talking to you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.